Para iniciar nossa conversa, convido ao palco o Dr. Jefferson Fernandes, que será o nosso mediador nesta tarde. Dr. Jefferson Fernandes é consultor internacional em saúde digital, curador do The Future of Medicine, diretor do Programa de Educação da International Society for Telemedicine and e-Health na Suíça, coordenador do curso Knowledge in Digital Health da Nova Medical School, na Universidade Nova de Lisboa, em Portugal, e vice-presidente da Comissão de Saúde Digital da Associação Brasileira de Medicina. Seja bem-vindo ao palco, Dr. Jefferson.
Muito obrigado. Boa tarde a todos amigos, colegas que aqui eu tenho a oportunidade de rever, colegas do cooperativismo. Eu ainda sigo ligado à Unimed Porto Alegre e é com grande prazer que eu estou aqui no Unimed Talks, nessa bela parceria do Future of Medicine, do Fronteiras do Pensamento, com a Unimed Porto Alegre. Eu também trago aqui as saudações do Dr. César Fernandes, presidente da Associação Médica Brasileira, que deseja a todos nós um excelente evento. A Dame Linda Patrick, professor Dame Linda Patrick, é uma das mais reconhecidas cientistas do mundo na área da longevidade e do envelhecimento saudável. Ela é professora emérita do University College of London, da Inglaterra, e também foi fundadora e diretora do Instituto de Envelhecimento Saudável do Max Planck Institute na Colônia, Alemanha. E as suas pesquisas pioneiras revelaram que é possível prolongar a vida saudável por meio de intervenções genéticas e nutricionais, inspirando novas opções farmacológicas, até para doenças como Alzheimer e Parkinson. Em sua carreira profissional, ela recebeu inúmeras honrarias, membro honorário de várias universidades, e também atualmente é membro da Royal Society e da Academy of Medical Sciences, tendo recebido o título de Dame Commander of the Order of the British Empire por suas contribuições à ciência. E a professora Linda é considerada uma das vozes mais influentes na busca por uma longevidade com saúde e propósito. E é uma referência na fusão global da biologia evolutiva, da genética, da fisiologia e da farmacologia, para entender como envelhecer bem e não apenas viver mais. Professor Partrigde, please, the stage is yours.
Thank you very much, Jefferson, for the extremely kind introduction. And also, I'd like to thank Ricardo Nicolay for very kindly arranging this visit, and Jefferson for the invitation to be here. And thank you all very much for coming to this talk and for tolerating it being in English. I don't think you'd enjoy it if I tried to give it in Portuguese. So, we're in a beautiful building, and here's an architect who I think is an outstanding example of healthy aging. It's Oscar Niemeyer, famous Brazilian, and he died in 2012, aged 104, and he maintained extremely good health well into his 90s. He continued to work, and only at the very end of his life did he start to get some of the indications of age-related problems. And generally, as we've heard in some of the introductory remarks, we've done a very good job of making ourselves live longer, improving our health, so that life expectancy increases. So this is quite an old article now, but I think it makes the point very clearly. So what we've got along the bottom is the year in which a birth took place, and up the y-axis, the life expectancy in the country that was the world leader at the time. So to start with, this tended to be the Scandinavian countries. These days, it's the Far East. And what you can see is this extraordinary 2.5 years per decade increase since the middle of the 19th century. It's really quite extraordinary. The horizontal lines are various people's guesses or calculations of where the trend would top out, but it didn't. It just kept going. And the causes were different at different times. To start with, we're talking mainly about neonatal mortality, mortality in young people from infectious diseases, bad food, unhealthy water, and so on. Starting in about the middle of the trend and now, it's mainly improvements in medical care, and the increase is happening almost entirely in older people. The situation in Brazil was mentioned just now. These are some of the data. So 1990 on the left, 2023 on the right. These are data from the World Bank, simply showing life expectancy at birth for women in pink, men in blue, and the average in green. And you can see the increase happening nicely there. Everywhere in the world, women live longer than men do, although as we'll see, those aren't healthy extra years. You can see the blip with the pandemic on the right-hand side. In most places, life expectancy is starting to recover, although in some places it's not clear. In the United Kingdom, there's been a distinct check in the increase, especially amongst men. There may even be a slight decline. So it's not clear where this trend's going in different places on the long term. But I think we have to celebrate that we've improved our health so much during our lives and that we're living longer. And it's leading to some amazing lifespans. So this is Jeanne-Louise Calment, a French lady, and as far as anybody knows, she is the world record holder. So she died in 1997 at the age of about 122 and a half. And I think nobody really knows what her secret was. She only gave up smoking when she was 117. It certainly was not a healthy lifestyle story. And this is quite typical of these centenarians. It's really not clear how they did it. This is the oldest living person at the moment. She's actually British. She's the first person to hold the record. And she's now 116 years old. Again, no clear lifestyle indications of why. So there is a genetic component to lifespan. It's maybe maximum 25% heritable, no more than that, probably quite a lot less. But mainly, we're talking about lifestyle factors here. And the typical lifestyle factors are the ones that you all know about, the ones that your mother told you. So if you do the proper epidemiological studies, what you find is that a healthy diet, particularly not eating ultra-processed food, plenty of exercise, good social life, good sleep, don't smoke, all the things that we know about. These are the major determinants of lifespan in the vast majority of the population. So there's a big public health fix that can be done straight away to keep people healthy as they age. But there are some knock-on effects of the fact that we've improved our health that aren't so good, and we've heard about one of them. These are what the data look like for the age structure in Brazil. So these data they span 1970 to 2022. And this is just a standard age structure pile. So you've got the different age classes vertically for men and for men in blue, women in red. And you can see the trend up to 2022 that the Brazilian population has increased a lot. The size of the whole thing has increased. But you can also see this bulge gradually creeping up from the initially very young population, which is now at about 40 to 44 years in the population. But it's going to go on up, as we've heard. And what that means is that things like dependency ratios, cost of retirement, and so on, all the things that you'd think about, are changing quite rapidly. And the big picture here is that we're just living much, much longer than we did in our evolutionary past. Very few people would have made it to 40. So what's being uncovered, despite the improvement in the living conditions, is that advancing age is the major risk factor for all sorts of things, subclinical and clinical. So this is a picture for the major diseases. So we've got cardiovascular disease, dementias, and neoplasms are the three categories there. And we've got 2 low, 2 middle, and 2 high-income countries. And this is disease incidence, somewhat scaled for the intensity of the data in the different countries. But what you can see is whatever the economic status of the country, you've got this steady increase with age. Quite a big difference in neoplasms for men and women, much commoner in men than in women. So advancing age is the major risk factor for most diseases. And it's leading to more than one disease in a single individual, multimorbidity, which I'm sure the clinicians in the room are very familiar with. These are data for Scotland. They're for men, as it happens. The women look very different, and this is a very typical global picture. So you've got ages along the x-axis up to 85, and then out of 100%, the proportion of the individuals that have got no diseases, one disease, two disease, and so on, as it gets darker blue. And what you can see is that by the age of 70, virtually everybody's got something, and most people have got more than one thing. So for geriatricians, this introduces all kinds of complications with treatments for multiple conditions, interactions between drugs, and so on. The other thing that is happening as lifespan increases is that health span isn't keeping up. So these are global data now. And what I mean by health span is the age at which the first age-related disease appears. And you can see globally the increase in life expectancy at the top between 2000 and 2019. And then in the pale blue lower down, you can see health span. And the gap between the two is growing. And it's greater for women than for men. Women have a longer period of ill health at the end of life than men do. You can see this here for the Brazilian data. So we've got lifespan on the left against year. You can see the blip with the pandemic on the right-hand side. The green's the average. And those are the healthy life expectancy data on the right. And by the time you get to 2021, the gap for women between health span and lifespan was 12.4 years, only 8.7 in men. So it really is a very big difference. The burden of disease is higher in women. So the way that I think most of the people who work in the kind of biology of aging field that I'm in look at this is the following. So what we have is lifespan in the past, where there was a healthy period and an unhealthy period. We've seen the increase in overall lifespan, and both health span and lifespan have increased, but that unhealthy period at the end of life has increased over that same period. And what we would like to do, ideally, is this. So for a given lifespan, what we want to do is to squash that red period at the end of life. You could view that simply as saying, we want to delay the age of the first age-related disease. But actually, if you ask people themselves what they value most about their lives, it's not necessarily just not having one disease. And the World Health Organization has come up with something which I think is a very interesting idea, which is intrinsic capacity. People's ability to function at home, to get around, to have a social life, to have good cognition, to be able to hear and to see people. It's a much more nuanced kind of definition. But whatever definition we use, what we want to do is to squash that period at the end of life where people are suffering a bad quality of life because they're old. So what are the prospects of doing that? I'm a biologist, and I was originally an evolutionary biologist, so I take a very biological view of this. And one thing that we can say straight away is that the aging process is extremely malleable. Some things don't age at all, and these are some of them. So these curious little creatures are a sea anemone, the little freshwater hydra, and a planarian worm. And as far as anybody can see, if you get these creatures into the lab and just look at their mortality rate and their reproductive rate, they just flatline. There's no indication that they get more likely to die or less able to reproduce as they get older. And one of the interesting things about all of them is they're basically bags of stem cells. They have extraordinary powers of regeneration. You can cut that worm into slices, and each slice will make a new worm. And it's because of these stem cells. And the stem cells can also give rise to the germline, the sperm and the eggs, and that's probably very important. So I think they're an existence proof that aging doesn't have to happen. And if you go out there in nature, there are some extraordinary lifespans, and they're not just explained by body size. This is clearly a trait that has a genetic basis and can evolve to happen at different rates in different kinds of creatures. So at the top, we've got, I think, the mammalian record holder. It's the bowhead whale. It was actually aged by the age of the harpoon heads that were found embedded in the skin of the animal, and they can live over 200 years. This Greenland shark, the vertebrate record holder, was discovered a few years ago. It just lives quite deep in the Atlantic, and it can live almost 400 years. It doesn't start breeding until it's 150, so extraordinarily slow life history. The little ocean quahog on the left is one of these bivalve mollusks, just lives on the floor of the Atlantic, and by isotope dating, they've been dated at 500 years old. And these two on the right I've put in simply because they're small, yet they have very respectable lifespans. So the naked mole rat is an extraordinary creature. They live in colonies underground. They're very social. But it's about sort of intermediate in size between a mouse and a rat, which each live 3 to 4 years if they're lucky. It can easily live 30 years. And I think the most remarkable is Brandt's bat on the bottom right. It's a tiny little bat. You can just see it sitting on someone's finger. It only weighs 8 grams. And it can live 40 years out there in nature. And actually, one of the growth areas in biology of aging at the moment is to sequence the genome of these creatures and start to try to figure out if they've got some interesting tricks for slow aging that we don't know about. But from my point of view, they establish the point that we can clearly alter the rate of aging a lot with genes. And if we could find those genes, maybe we could target them to improve human health at the end of life. So as far as genetics of aging in humans go, there are some extremely sad, but happily extremely rare cases of at least partially accelerated aging. So these are the progerias. 2 of them are shown here. The most extreme is the one on the left, which is Hutchinson-Gilford progeria. So those children are born quite normal and stay that way till the age of about 18 months or 2 years. I know there's a pediatrician in the audience. I hope I'm getting this right. But then you gradually start to see the symptoms of the condition. So they start to lose their hair, their subcutaneous fat. They get stiffening of the joints. And they typically die in their teens of either cancer or cardiovascular disease. Less severe is Werner syndrome on the right, so that they're normal usually till they're in their teens, but eventually usually die in their 40s or 50s of cardiovascular disease. And these are very sad conditions, but I think they do also tell us something quite important mechanistically about the biology of aging, because they both involve the way that the genetic material in the nucleus of cells is controlled. So the progeria is a mutation in a gene called lamin A, and it makes a protein that's in the nuclear envelope in the cell. And so the chromosomes and the DNA in the nucleus become disorganized. The spatial arrangement goes wrong, the wrong things interact with each other. So what it's telling us is that integrity of the genome during aging is really important. The one on the right is a mutation in a gene that encodes a DNA helicase. So this is a molecule that winds DNA during cell replication. So again, it's telling us that fidelity of the genetic material is really important. But of course, what we really want is to find the genes that can improve health during aging, not destroy it. And for that, these have been the real workhorses. So these are the standard laboratory model organisms, budding yeast, the nematode worm, Caenorhabditis elegans, Drosophila in the mouse, where I do quite a lot of my own work. And they've been so powerful because they're easy to keep in the lab, they're cheap, you can do experiments with them quickly. And there's very, very strong evolutionary conservation of biological mechanisms. So much that we've learned about genetic transmission, control of gene expression, the way the nervous system works in the multicellular organisms, has turned out to be directly applicable to human biology. So they've been amazing engines of discovery. You can often take a gene from one of these or from a human and put it in another species, and it works just fine in the new location. But it wasn't clear that that was going to be true for aging. They live for very different lengths of time. I mean, typically a worm will make 3 weeks, a fly about 3 months, and a mouse about 3 years. And they encounter very different challenges as they go through life. But eventually somebody very clever stopped overthinking it and just did the experiment. She was working with the worm, and what she did was to ask, can I isolate genetic strains of worms that live for a long time, longer than the wild type? and dramatically she found that she could. So this is the famous DAF2 mutant, and you've got a survival curve here, so the controls are the open boxes, and they hit the deck at the age of about 35 days. But you can see the mutant in the dark boxes is incredibly long-lived relative to the controls. And very importantly, this was an extension of healthy life. They were wriggling and healthy long after all the controls were dead. It's not an extension of the pathological period of life. And it took a while to figure out what this was, but eventually, to everyone's surprise, it turned out to be a mutation in the worm gene that makes the insulin-like growth factor receptor. So it wasn't even suspected that that was present in a worm. And of course, much more familiar in the context of control of metabolism and growth and wound healing in mammals. But here it is in the worm, affecting their lifespan. And basically what this is a good times network. If there's plenty of nutrition, there are no stresses impinging on the animal, then the signaling is going at a high level. The various transcription factors it controls are withdrawn from the nucleus, and it goes in for costly activities, reproduction, metabolism, growth, and so on. But if there are problems, then it turns on genes that are involved in stress response. It's a very ancient, evolutionarily conserved pathway. And one of the important things about these mutants was that it turned out not only could they improve the health of the wild-type worm during aging, but they could also rescue the pathology associated with worm models of age-related diseases. So I'll just show you one of these. This is a worm where the A-beta peptide, which is so important in Alzheimer's disease, has been expressed in the muscles of the worm. And you can see it's completely paralyzed at the back end, and the front end is twitching quite feebly. This is what happens if you combine that mutation with the DAF2 mutation. So here's the Alzheimer's worm again. But now we've got a worm that's wriggling perfectly normally. And this was true for other worm models of age-related diseases, including some cancers. So I think people were quite struck by this finding, but of course there was the possibility it was just a long-lived worm, which is not particularly useful, but it did turn out to be evolutionarily conserved. So subsequently, people looked at similar mutations in the fly and the mouse. So Chico is the single fly gene that encodes an insulin receptor substrate, which sits just below the receptor, the DAF2 product, and the mouse on the right has lost both copies of insulin receptor substrate 1. There are 4 in the mouse. And you can see that in both cases, you get a long-lived animal. Again, the phenotyping has been done to look at health during aging, and there's a really quite remarkably broad spectrum improvement in health. So just that particular mouse, the IRS1 mouse, these were some of the features that turned up. So this is as the mice age, the mutant mice age, they've got better control of their glucose homeostasis, despite being a mutant in insulin signaling. They've got a better immune profile, more naive T cells, better motor function, they're more agile on a rotating rod, and they get less osteoporosis. You can see the cataract on the mouse on the right, that's one of the control mice, and also this ulcerative dermatitis that about 40% of that controlled strain typically get. We never saw a single case in the mutant mice. So I think what's interesting here is this really broad spectrum improvement in health from a single mutation. Clearly, we'd like to know if this is applicable to humans, and the main way that that has been tackled is through population genetics, looking for associations, sometimes candidate genes, so taking the human relatives of the genes that have turned out to be important in the animals and saying, are genetic variants associated with long life, or just taking a more genome-wide black box approach and say, if we look at really long-lived people, which mutations seem to be associated with that long life. And if you do that, these are some of the papers that have come out. There have been signals in IGF-1 and TOR signaling. But the one to me that really is important is that FOXO3A, which also turned up in this meta-analysis of all the studies that have been done. And the reason that FOXO3A is important is it's a transcription factor, so it turns on and off the expression of genes, and it's controlled by insulin IGF signaling. So when signaling's active, FOXO is pulled out of the nucleus. When things are getting tough, it goes into the nucleus, and it's the transcription factor that controls these stress response genes. There's a single FOXO in the worm and in the fly, and it has to be present for decreased insulin IGF signaling to increase lifespan. So if you knock it out, the animal's alive, but it no longer extends its life with the insulin mutants. And we've actually got recent evidence that that's also true in the mouse. And it's consistently turning up as important in the human studies. So this is just one line of research which I happen to have been involved with, which has been going on really over the last 20 years or so. But plenty of other work's been going on in parallel, as a result of which we really know a lot about the aging process now and its underlying mechanisms. So they've become known as the hallmarks of aging. They're shown here on this wheel, just to very quickly whistle through them. Top right, we've got basically 3 genetic mechanisms, so control of the genetic material I've already mentioned. The ends of chromosomes contract during cell division, and that can be a problem if telomeres become eroded. There are molecules that stick on the DNA or stick on the proteins that are wound around the DNA and that control gene expression, and that becomes dysregulated during aging. Within cells, proteins are obviously the key effectors of the cells, and frequently cells lose control of the protein domain. Either the degradation or the production becomes impaired. We've already heard about the nutrient sensing network, insulin, IGF, and TOR. The mitochondria in the cells, really important for energy generation, but also huge metabolic crossroads, become impaired. Cellular senescence is something that really has come out of the aging research field. So during development or wound healing, senescent cells are produced. So they're very biologically active. They secrete a lot of cytokines, proteases, and they can destroy tissue. They're responsible for the tissue rearrangements that happen during development and during wound healing. So you need them. You don't want to get rid of all of them. But normally the immune system comes and removes them when the process is resolved. That goes wrong during aging. So they accumulate in tissues. And there's some very nice experimental work with mice now showing that if you remove senescent cells, either with a drug or genetically, you can improve tissue health in the mouse. And there are a number of clinical trials going on off the back of that now. Stem cells we've already heard a bit about. They misbehave during aging. They either fail to differentiate when they should, or they misdifferentiate, and that can cause big problems within tissues. Altered intercellular communication is talking particularly about the immune system. Inflammation during aging is a major cause of problems, but also immunosenescence, where the immune system doesn't do what it should. And it's clear that the systemic environment fills up with damaging molecules during aging, and their removal can actually improve health. So these are the basic mechanisms. They're not independent. They interact with each other. But what's really important is that they're all present in the etiology of multiple aging-related diseases. These are the guys that are initiating the disease processes that wind up with the aging-related diseases that we see. And one major area of growth at the moment is trying to tackle these hallmarks individually to improve health. Some clinical trials, some of it's still at the animal research stage. So I'll just show you 2 examples that I think are particularly impressive. So sarcopenia, muscle weakness, obviously a big problem during aging. And Helen Blau has discovered that typically after exercise or muscle injury, prostaglandins are released. And during aging, an enzyme that degrades prostaglandins for some reason increases in expression. It's not clear why that happens, but it means that the normal prostaglandin response is not there, and the muscle stem cells don't divide. There isn't the normal repair process. And she's now got in stage 2 clinical trials a small molecule that inhibits that enzyme. So she's already done all the preclinical work. It's safe, and it's going through. And what she sees in mice is that it as I say, restores stem cell function. It restores the mitochondria, the fiber structure, and the strength and size of the muscle increases. This is in old mice. So that one, I think, is really going through. A little bit further back is a drug called rapamycin, sirolimus, which many of you in the room will know, and I'll say more about it in a minute. But one thing it does is to induce autophagy, which is this process of cellular recycling of garbage, damaged macromolecules, and organelles. And what it can do in animals is to restore the functional integrity of the intestine, which breaks down during aging. You start to get gut leakage, bacterial products go into circulation, probably responsible for quite a lot of the age-related inflammation that we see. And rapamycin can reduce that and restore gut integrity. And that's being used in multiple clinical trials for age-related conditions in humans. And as I guess you're aware, that this is a real growth industry area. There's an annual conference in Copenhagen, Aging Research and Drug Discovery, which I go to sometimes. I went last year. And these were just the sponsors of that meeting. So a lot of them are, worried well longevity clinics, but a lot of them are also doing serious basic and translational scientific research. So everywhere, I think this is something that is growing. But my own interest is in something a bit different and a bit broader, which is the idea that because these aging hallmarks are each present in the etiology of multiple age-related diseases, maybe what we could do is tackle a single hallmark to delay or prevent multiple age-related diseases. So the idea here, to parody it, current situation at the top, where we basically know that the mechanisms of aging are leading to these diseases, we wait for them to crop up and then typically try to treat them once they've occurred. The geroscience idea is that rather we could acknowledge that these mechanisms are there and leading to disease, but we'll tackle the underlying mechanisms themselves much sooner to try to delay or prevent the things that go wrong later on. So one obvious question is there any evidence from human data that that approach might work? Is it true that individual aging hallmarks do contribute to the etiology of a group of diseases? And is that group of diseases more likely to occur in real patients? In other words, are patterns of multimorbidity partly explained by their common association with these hallmarks of aging? That's something we've been working on at UCL, and these are the two key players here. So Valerie Kuan did her PhD analysing data. She had a remarkable history. She's also a GP, and before that she was a trader on the stock market floor in London, and before that she was an actuary. So she knows how to analyse data. And Arun Hingorani was her PhD supervisor. He's a cardiologist at UCL. And what they've done here is to ask, well, which diseases are aging related? And so they've got 3 million National Health Service records, both primary and secondary care admissions. They needed a lot of cleaning up. But what you're looking at is the rate of incidence of 278 high-burden diseases. And Val's done a cluster analysis here, and they fall roughly into 9 clusters. And the first 4 of them are clearly aging-related. So cluster 1 actually hardly has any diseases in it. It includes delirium. Cluster 2 is mainly cardiovascular disease. Most cancers are in cluster 3. And cluster 4 is quite a ragbag, but there are a lot of intestinal disorders in there. But there are 207 diseases in those 4 clusters. So most diseases are age-related in their instance. So then we looked at which hallmarks are associated with these diseases. So this AI comes in, machine read nearly 2 million abstracts in PubMed for a mention of these 207 age-related diseases and a co-mention of 1 or more of the 9 aging hallmarks, and then look for co-occurrence. I mean, that's obviously statistically tricky, but can be done. And that enabled us to define the top 30 of those 207 diseases that were associated with each hallmark. And that's what this looks like. There were actually 83 diseases that were thereby pulled in. They're listed down here, and you've got the 9 aging hallmarks at the bottom, and the depth of the colour indicates the strength of the association. And a lot of things that you would expect came out, so loss of proteostasis with neurodegenerative disease, dysregulation of the genome with cancer, those sorts of things all came out, mitochondria and nutrient sensing and metabolic disease. And there are some interesting patterns here. Breast cancer, for instance, is associated with all of the hallmarks, whereas in this data set, some are associated with only one. But the really critical question is, all right, we've got these associations, there could be all kinds of reasons for that, bias in the literature and so on. Do they actually have any explanatory power for the co-occurrence of the diseases? So for that, we did a network analysis. So what we've done is to build a network for the 30 diseases associated with each hallmark. And we ask, is that network tighter than you would expect from the overall network for those diseases? So it's asking directly, are the associated diseases more likely to be present together in the same patients? And the answer was that they were. And this was particularly true for the nutrient sensing, mitochondrial dysfunction, cell senescence, stem cell exhaustion, and the altered intercellular communication. And Helen Fraser, who did this work, actually divided the data into 4 decadal networks, 50 to 60, 60 to 70, and so on. And the associations were stable right across. So I think potentially this approach does have legs, and it also could, to some extent, help with dealing with the more public health aspects of ill health during aging, the ones that are associated with socioeconomic inequality, which is, of course, the major cause of variation in ill health during aging. And again, it's a real privilege to be at UCL because of colleagues. There are fantastic epidemiologists and demographers there. So we've recently been collaborating with Mika Kivimaki, who's the professor of social epidemiology at UCL, to simply ask, is there an association between social disadvantage and acceleration of the diseases that are associated with particular aging hallmarks? So he has a number of cohort studies that he's involved with. Social deprivation can be defined in many ways. It's really a composite measure. But he looked at education, paternal occupation, the deprivation level of the neighborhood in which the person lived, occupation, family income. And 66 of those 83 hallmark-related diseases were associated with social deprivation. The 30 most strongly deprivation-related ones actually were associated across the board with multiple aging hallmarks. But particularly important seemed to be stem cell exhaustion, nutrient signaling, intercellular communication, and mitochondrial dysfunction. So again, it argues that these aspects of social deprivation may be functioning through these very basic biological mechanisms. Finally, I'd like to come on to my own current interest, which is in the potential of either developing or repurposing drugs to try to target these aging hallmarks. And we had a discussion about this earlier. Would you be prepared to take a drug to maintain your health during aging? And I think my answer to that is, well, a lot of people already are. This is particularly true for preventing cardiovascular disease. Millions of people are taking statins to prevent atherosclerosis. It's been one of the most successful drugs ever, as are drugs that lower blood pressure. So these are trying to prevent conditions that have not yet occurred and are generally quite acceptable. And of course, more recently, the GLP-1 agonists are having the most amazing effect. So I think there would be take-up of safe drugs that kept people healthier for longer. And for that reason, we've been doing some work on them. So back to the nutrient sensing network now. We've got our hallmarks of aging and dysregulated nutrient signaling. And this is what this little signaling network looks like. If you grossly oversimplify it and base it on the fly, the mammalian thing looks much, much more complicated. But the basic story here is you have the receptor, which I've already mentioned, the receptor substrate. And then there's a bifurcation of the signaling pathway. One branch on the right goes to FOXO, which I've already mentioned. And the one on the left is the RAS pathway, which will be familiar to many of you because it's great important in cancer. And you can see the TORC part of the signaling network in the center, which is targeted by rapamycin. So we're particularly interested in two drugs. One is rapamycin, and what it does is to disrupt that TORC1 complex. It's a protein complex, and rapamycin stops the complex from forming correctly, and so reduces signaling. Its normal on-label uses are to prevent transplant rejection, particularly kidneys, to prevent restenosis after cardiac surgery, and as a cancer chemotherapeutic. And it's long off patent. Trametinib is a GSK drug that's a MEK inhibitor. So it was originally developed to treat melanoma. It's now used for a number of other cancers, often in combination with another drug. And it also signals down to a transcription factor. So why are we particularly interested in these two drugs? Rapamycin became famous because it was the first drug that was clearly shown to increase lifespan in mice and to improve their health during aging. So it was actually discovered on Rapa Nui, which is Easter Island, hence its name. It's a natural product. It's produced by bacteria in the soil there. And you can see this nice dose-dependent increase in lifespan in the females and the males. This is at a much lower dose equivalent than is used clinically in humans. And most aspects of health during aging are improved. It does have side effects, even in mice at these very low doses. But we have a lot of indication with this drug that the timing of treatment is really very important. You can give it very intermittently and get the effects of chronic treatment. And also it induces the most extraordinary memory effect. So if you give it to young mice or flies, withdraw it and wait until they start dying, you still see the increase in lifespan that you would get with chronic treatment. So it's somehow really kicking the system into a different phase. It's a very, very interesting drug. And a lot of clinical trials are being done with it in humans for geroprotection rather than for single disease. And this is one of the really early ones. So this is Joan Manick, who's now at Altos, one of the companies that you saw on that earlier slide. And what she did was to build on an earlier mouse study that had shown that if you pre-treat mice with rapamycin, withdraw the drug, let them clear it, and then immunize them against flu, you get a much bigger immune response to the immunization, and they're protected against a flu challenge. So Joan simply wondered whether this might also be the case in humans. So she did a very similar experiment where she took a group of older people who were about to be immunized, gave them the drug, looked at the immune response and infections, and what she found was that there was a positive response. So the immune response was stronger, and they were less likely to get validated respiratory viral infections in the ensuing winter. So I think this is a lot of the way that these drugs will come in through tests, with a single indication, as in this study, although I'll say more about that in a minute. Trametinib we became interested in because of work that we'd done that I don't have time to describe in any detail, but it was with Drosophila. And although the RAS pathway has been sitting there in plain sight under the insulin receptor, because of the information about FOXO, I think everybody just assumed that only the other branch of that signaling network is important. But we could show in Drosophila that if we prevented the connection of Chico with the RAS pathway genetically, we got a fly that was just as long-lived as if we knocked out Chico. And we could also show that this drug, trametinib, which is, of course, developed for humans, works perfectly well in flies, and it extends their lifespan and keeps them more mobile and other indications of healthy aging. So we got this drug, but only in flies. And we were lucky we got the funding to look at it in mice. Unfortunately, we were a bit overcautious, but nevertheless, this is what we found. Because it had never been used in this way in mice before, we were very, very keen to ensure that we didn't induce side effects. And I think we went too low, and we will go higher. But what you can see is that in both sexes, there's really quite a nice, consistent increase in lifespan. It seems to be a bit larger in males than in females. So it's looking as though it's geroprotective in mice. And what we could see in these trametinib-treated mice was a big reduction in age-related inflammation. That was the most obvious thing. But the other thing that we'd shown in Drosophila was that if we gave them rapamycin and trametinib together and at the dose that maximized the increase in lifespan when they were fed singly, what we saw was basically an additive increase in lifespan. We saw a much bigger increase in lifespan with the two together, even though they were at their maximum effect. There could be a lot of reasons for that. They may inhibit the same targets more strongly. They may bring in different targets from each other. There's a lot of feedback in these signaling networks. It could be preventing that, preventing the system from fighting back. But whatever, they clearly work very well together. So we had a look at this in the mice. And you can see here the two that you've already seen, the mauve and the green, which is the trametinib. Red is rapamycin and Arhans, intermittently fed to the mice. And the orange is what happens when we give the two drugs together. We don't know what the maximum of either of these drugs would be for increased in lifespan. But you can see that by mouse standards, we've got a really huge increase in lifespan here. It really does extend it. And we saw a marked reduction in age-related inflammation, again, particularly in the brain. Interesting, because trametinib does not go across the blood-brain barrier. So there's something interesting going on there. But also reductions in tumours, particularly in the kidney and in the spleen. So, we're hoping to do a lot more work on this. The one thing I would say is that trametinib, I think, is about to become off-patent, and rapamycin already is. And there's a bit of a valley of death here, because if you want to do clinical trials with off-patent drugs, certainly industry are not going to pay for those trials. Aging is not recognized as a disease. It's not an outcome for clinical trials. So you have to go for particular indications. And if you're interested in aging, you want to go for a broad range of indications. And we're talking about prevention. So a lot of people over a long period, very expensive trial. So even though the indications are there, that these could be quite powerful public health measures to keep people healthier as they age, actually getting there, I think, is going to require either public funding or charity funding. Otherwise, it's not going to happen. So that's a discussion. But it's clear that if somebody did pay, the economic benefits would be huge. I mean, there are various ways of cutting this one in terms of looking at health economics. But if you could even put off the first aging-related disease for a year, you'd see a huge economic benefit of doing so. So, to summarize what I've said, aging hallmarks lead to age-related diseases, and they can be targeted with both lifestyle and drugs. So interventions like dietary restriction and exercise clearly go through one or more of these hallmarks. Targeting the nutrient sensing network with a combination of drugs can be very effective in ameliorating aging in mice. We would really like to do the experimental medicine and clinical trials with them, but who knows? And these findings point to at least the possibility of a broad-spectrum preventative intervention for the diseases of aging, with benefits for both quality of life and the economics of healthcare. So I'm going to finish there. I'd like to acknowledge our funders and our model organisms, and you for coming and listening to the talk.
Thank you very much, Professor Partrigde, for your fascinating and insightful lecture.
Thank you.
Lots of things to learn, to understand, and in the end, to translate into a better health care for all of us before you get aged, but also during our aging process. Muito bem. Bom, I'm going to start asking something that you have touched in your lecture, but it's, I mean, from your work, you just demonstrated that aging is something very malleable. And for those things, let's go to interventions that might be used in humans. From your view, what would be those that you think are more prone or they have a greater probability of being able to be translated from the labs to clinic and to humans? which ones would you bet will be coming for us?
It's an incredibly important question. And I picked this up mainly through going to aging conferences because I'm not a clinician myself. One of the ones I was very interested to learn about recently is trying to use some of our knowledge of the way that tissues age to improve the health of organs for transplant. So a lot of this is going on in Barcelona at the moment, and they're looking at senescent cells in organs that are about to be transplanted. I mean, already the surgeons are extremely good at eyeing up a potential transplant organ, saying whether it's good or not. But the question is, can we improve on that by removal of senescent cells? At the moment, they're doing most of the research actually in pigs in Barcelona, but that one's creeping close. I think the sarcopenia one is also creeping close. There've been a lot of disappointments with cellular senescence. There's a particular cocktail of senolytic drugs. So these are drugs that selectively kill senescent and not normal cells. They're pretty effective, but they're rather toxic. So the thought is that they could be used either for cellular senescence in conditions where it's contained somewhere where the drug won't spread much, like the eye or a joint. So there's been some work going on there. And also pulmonary fibrosis was the first safety trial because there's no other treatment for that condition. At the moment, the drugs are so toxic that I think probably the existing wave are not going to go forward. But of course, we've got all this wonderful AI-aided drug discovery now, both in terms of identifying targets and identifying molecules that don't necessarily even already exist that could interact with those targets, including ones that have previously been thought to be undruggable. So I think there's a lot of potential there.
Okay.
But it's not coming into the clinic tomorrow, I don't think.
Yeah, it takes time.
Yeah.
That's why, because not only aging, but aging well, it's important not only for the individual or his family, his friends, but also for the health services and health systems. Decreasing the chance of having heart diseases, dementia or cancer is important, so people would not only live better and longer, but also it decreases the cost of the healthcare, which is increasing a lot. Unimed is one of the largest health operators in Brazil and is really pushing to strategies such as value-based care to try to reduce not only the cost, but also to improve outcomes of patients. And so we have lots of initiatives that goes both in the same direction. Let's talk about, let's say, personalized or personalized medicine, lifestyle medicine, and the use of artificial intelligence. So you mentioned now artificial intelligence for the discovery of new molecules without needing to have to go through the long process of preclinical activity. Besides that, what else do you think artificial intelligence can contribute in the understanding and also in the future interventions for long life?
Well, it's coming in all kinds of different ways. I mean, I mentioned those patient health records. What I've talked about is just scratch the surface. And, the basics have been done, age-related, sex-related, ethnicity, and so on. But what artificial intelligence is really good at is looking at a large pile of data and finding patterns. And I think a lot of its input is going to be into epidemiology, into analysis of data, getting a much better grip on what's a risk factor for what. If somebody's taking drug A for condition B, does it also actually protect them against condition Q? There's been very little of that kind of research so far. So I think there's a huge amount of just human data work that's waiting to happen. robotics, in which there's a lot of AI. It's not really in people's homes yet, but there's an awful lot of work going on developing robots that can help people to stay in their own homes and to be safe and functional when they're there. That's the most recent one I saw about was one that could actually physically support people with sarcopenia and help them with their balance. So, you know, it sounds almost space age, but that really is coming. The trials with that are being done. Drug development's another really important one that I've already mentioned. So it's going to be everywhere.
Okay, right, thanks. We have a lot of questions from the audience, so I start with the one from Livia. How the United Kingdom deals with the lack of social support and family support for long-living persons? Many times aged people stay without their relatives and friends for the basic care. How is UK dealing with that?
Badly. It's a real problem. There have been successive governments, I'm getting into politics now, have tried to rationalise the care system for older people. Because at present, the situation is that if somebody goes into care and essentially they have any assets at all, those assets have to go on the care. So, you know, people are selling their houses and to get themselves looked after. There's a huge shortage of places in care homes, and there's a huge shortage of people willing to work in care homes because it's so badly paid. Often people who are caring for old people are being paid less than the people who are stacking the shelves in the supermarket. So I would say that there's just a massive policy failure in the UK, and I really hope things are better here.
Right. In the same approach, Leandro Firmi asks which way the economic and political changes and in the models of financing, health financing, dialogue with the medical advances and technological advances that are redefining the limits of human longevity.
Yes, I think that really is partly taken care of by the fact that my aim, I'm quite clear, is that I want to compress that period of ill health and loss of function at the end of life. And that should not pose economic challenge. It should only lead to economic gain and also increasing the quality of life for people to have a less bad time at the end of their lives. But obviously the growing period of morbidity and disease at the end of life is a huge challenge for economic systems. And you know, medical care systems worldwide are struggling to cope.
Mm-hmm. Right. And Adriana asks, what is your view regarding the effect of new molecules such as Ozempic and Wegovy in the process of aging of the population?
Well, I think it doesn't matter much what age class you're looking at, actually. I mean, a lot of the people who take the GLP-1 agonists are young, and they're doing it to lose weight. But all the indications are that maintaining a healthy weight is really important. If people can do that through their lives, it's one of the single most important things that they can do to ensure that when they hit old age, they are in reasonable health. The other one that's huge, and where I think it almost requires public health measures, is exercise. So one of the reasons I really admire societies in the Far East is partly that they value old people and they look after them well, but also that there's just a culture that promotes exercise. So in China, you can easily see a square that's in the morning, it's full of old people doing Tai Chi or some other form of collective exercise. I was telling the journalists earlier, I even read a piece about 2 weeks ago about South Korea, that they're starting to open discotheques for older people. So that these very simple cultural things that mean that people get out of their chair and get the exercise, these would be serious public health measures. But I think our politicians and policymakers, they just don't take them seriously enough.
Right. Paula Sa, journalist, she asked a lot of questions. I just I'll pick up one of them, so we have a lot of people asking. And he brings an interesting question. You have these, interventions that are being in research to deal with the aging, but how to do it without compromising health? The question of the balance between side effects, let's say for drugs, and the benefits.
Well, that's obviously an absolutely key question. It's really important. And I think one of the reasons that statins in particular have been so successful is that for the vast majority of people, they don't have side effects. And I think if a lot of people are going to take a drug over a long period, that has to be true. I mean, obviously, you titrate benefits against harm. If somebody's got a killer disease and there's a drug that can bash that killer disease on the head, then they're going to be prepared to tolerate more side effects to do that. But if you're really talking about prevention in healthy individuals, then the drug has to be essentially free of side effects.
Right. Going back to research finances or supporting sponsorship. How NHS is participating on that? How much it's putting money on this sort of research in UK?
Rather little, actually. The research funding system in the UK is going through a bit of a spasm at the moment. The main government funder, UKRI, is coming under increasing government inspection. I mean, this is something that happens in cycles in all countries, I think. But I think the government at the moment are concerned, well, obsessed with productivity, and they want the research that's done to feed into UK PLC. They'll tolerate a certain amount of basic research, but not much. And both with the public funder and with the other major biomedical funder, which is the Wellcome Trust, they don't have the ageing population as a research priority. And it's an area that transcends a lot of different areas of research where you really need a coordinated push. So in the United States, they've got the National Institute of Aging. I'm involved quite heavily with an EU-funded project and, as an assessor in Italy, and they are trying to set up a National Institute of Aging there. And I think either, by coordinating different parts of the country or by actually being physically in one place, that's what you have to have, is all the different bits of the picture from the highly molecular through to the epidemiological in one organisation. And we just don't have that. We have huge strength, but it's really fragmented.
Right. One question from Liège. Are there studies regarding the environment toxicity and dementia and longevity problems?
Yes, that's a very sad topic, I think. I've just reviewed a book by somebody called Topol, who wrote a book called Super Ages. And there's a whole section in it on environmental toxins and their increase. And I think they're creeping in and we're starting to see their effects. I think the most striking one at the moment is the increase in colon cancer amongst young people. And really at the moment, nobody knows why, but it's really obvious. So in old people who are the most vulnerable to cancer, the incidence is actually going down, the age-specific incidence. It's gone down really steadily and continues to do so. But it's going up in the young. And that pretty well has to be some environmental effect that we don't know what it is. So I think we're starting to see the tip of that iceberg of what we've done to our environment. And it's not going to be pretty.
Mm-hmm. Right. So Carla Braga asks, she comments regarding the delay that we're going to have for having drugs that could be really used daily or used for people. And maybe it would be nice for you to repeat what strategies do you suggest for healthy longevity?
I think that, honestly, they really are the ones that your mother told you. Maintain a healthy weight. The composition of the diet's really important. And it probably changes a bit with age, actually, the right thing to eat. That one size doesn't fit all. So sarcopenia in older people probably means that they should slightly elevate the amount of protein in the diet to try to encourage muscle repair and maintenance. Diet's really important. When it's eaten is really important. There's quite a lot of evidence now that it's important to confine eating to the active period. So one of the reasons that shift work, working through the night, may be so detrimental to the health is that, obviously if people are running around working in the night, they're going to eat. And that means that they're eating in what would normally be the sleep period. And there's a lot of evidence that that's actually harmful. I mean, if you look at patterns of gene expression in a person or a mouse in the day and the night, you would think you were looking at a different organism. And there's a reason for that. The day period is when food's consumed, activity happens. At night, repair processes, detoxification, particularly in the brain, but also in other parts of the body go on. And if you start to mess that up, then, health goes wrong. So there's a lot with diet. I've mentioned exercise, obviously. Sleep's really important, good quality sleep. Not doing the bad things, the smoking and the too much alcohol. Good social life, good psychological health. Just all the things we know.
Right. Okay. What educational initiatives do you think would be interesting to train physicians or to qualify physicians to tackle all these issues of longevity? How much is this being taught in graduation course or in the medical continuous education?
I think that's really interesting. So one of the things I and my colleagues have pushed for quite hard at UCL is a bit of a change in the medical curriculum, because a lot of medical students come in thinking that they're going to be pediatricians or deal with young people. And actually the vast majority of the people they're going to see, almost whatever kind of speciality they go for, are going to be old. And I think they just don't realise that. Geriatrics in the UK anyway is still a coal-face science. It's patient-facing. There are very few clinician scientists in geriatrics and very little training during the student years. I think they're just not prepared for what's going to hit them, and they don't know how to think about it. So I think there's a lot that could be done with the medical curriculum. I don't know how it is here, but that's how it is in the UK.
Right. The field of longevity, and not only the field of longevity, is ripe with both hype and misinformation, myth and fake news, and a lot of people are making money with it.
Yep.
What patients and doctors should look for to identify ethical and evidence-based longevity care?
Needs to have been through a clinical trial. Needs to have been quality controlled. I mean, I'm always amazed at the things that people will put in their mouths. I mean, if you go into a standard drugstore, there are shelves of stuff for which there's absolutely no clinical evidence at all, and some of it may be harmful. I find this quite extraordinary. Think treatments, whether drugs or other kinds of treatments, they have to be based in evidence. I mean, I think the Hippocratic Oath, isn't the first line, do no harm? It's really important that treatments are evidence-based.
Yes, I was a few weeks ago listening to an Eric Topol conference, and he, in the Q&A session, he was asked his opinion regarding all these things, people writing books, suggesting people to buy things and so on. And he was asked, what do you think about it? And just said, I don't believe in people that write about longevity or well-being that sell anything for their readers. So this is one way of...
That's a good answer, yeah. And his book, I think, is well worth reading. He isn't selling anything. And he really talks about this fringe area, the fact that the subject area attracts a real difficult fringe of charlatans, basically. And he doesn't take any prisoners. I think he really goes for the evidence base.
Okay, so for the sake of time, I just ask you one last question. What message would you like to leave for this audience? Or messages?
Well, lead as healthy a life as you can. And I hope by the time that I see many young faces in the room, I hope by the time that you're facing the aging process, there will be a lot more things that have come online as a result of the work that we've been doing. Maybe too late for me, but I hope it's there for you.
Thank you very much, Professor Linda.